Alright, hi everyone. Today we'll be going over the IGCSE Edexcel Human Biology paper. So this is paper 1 from June 2022. And for this paper you are given an hour and 45 minutes. So this paper tests you on the whole syllabus. So you've got multiple choice questions, you've got data type questions, short answer, and also long answer questions as well. All right, and the total mark for this paper is 90 marks, and this paper constitutes 50% of your entire grade. All right, let's start off with the first question. So this is a diagram of the molecule of a DNA, and structure X, which is this, is the backbone of the DNA molecule. So it says which of these form part of the backbone of DNA. So if you recall, this is what the backbone of DNA looks like. So you have your phosphate, which is the circle, which are the circles here, followed by the sugars, and it just goes on. Okay, so the answer here would be sugars. All right, next part. What name is given to these structures labeled Y? So Y here are the bases. So these are the bases, and there are 4 bases for DNA. We've got A, T, C, and G. All right, describe how the 2 backbones of the DNA molecule are joined together. So the 2 strands are joined together by, number 1, hydrogen bonding. So remember that there are hydrogen bonding between the complementary base pairs. Between complementary base pairs. All right, and the base pairs for this, you don't need to write this in your answer, but the base pairs are A pairs up with T, or adenine pairs up with thymine, and cytosine pairs up with guanine. So this is how I personally remember it. So A, the letters A and T are both pointy letters, so they pair up together, and the letters C and G are curvy letters, so they get paired up together. All right. State the name of the part of the cell where most of the DNA is found. That would be the nucleus. All right, next part. DNA is a nucleic acid. RNA is another type of nucleic acid. Give 2 differences between the structures of DNA and RNA. Okay, so asking for the different structures. Oh, let me just underline that properly because that bothered me. Okay, so one of the differences is actually in the name. So the D in DNA stands for deoxyribose, and that's the sugar that is inside of a DNA molecule, whereas the R in RNA stands for ribose. So that's one of the differences here. So you can say RNA contains ribose, whereas DNA contains deoxyribose. Okay. So another difference that you need to know is that DNA is double-stranded, right? So it's in a double helix, whereas RNA is single-stranded. So RNA is single-stranded, and whereas, oh, sorry, this is supposed to be a dash there, whereas DNA is double-stranded. Okay, another difference as well, because it only asks for 2 differences, but another difference that you need to know are the types of bases that it contains, right? So if you recall, for DNA, the bases are A, T, C, and G, but for RNA, the bases are A, U, C, and G. So DNA contains thymine, whereas RNA contains uracil. So that's another difference as well that you need to know. Okay, next part. There are different types of RNA. Which of these are two types of RNA? So the answer for this is D, your mRNA and your tRNA. The M in mRNA stands for messenger. So the mRNA is actually made in the transcription process, right? Because you have transcription and translation. So mRNA is produced in transcription, whereas the tRNA, so T for tRNA stands for transfer. And what is it transferring? It's transferring amino acids to the ribosome so that translation can occur. All right, let's go to part C. DNA is formed from molecules called nucleotides. So here's a diagram showing a single nucleotide. All right, name the parts. R is the phosphate. S is the sugar. Or you can say deoxyribose as well, because it is a DNA nucleotide, right? And T, which is this rectangle here, is the base. All right, question 1 is done. All right, the graph shows how a person's heart rate changes with exercise. So here we have a graph. The y-axis is the heart rate in beats per minute, and the x-axis is the time. So this is a person exercising. All right, determine the difference between the maximum heart rate and the resting heart rate. Okay, so if you can see here, this is the resting heart rate. So this is before the person is exercising, and this would be the maximum heart rate. Okay, so we can see here this is the resting. So if this value represents 70, then this would be 72. And the maximum heart rate, so if this line represents 150, it's 246, so that's 156. So we would do 156 minus 72, and that would be 84. So the answer for this would be 84 beats per minute. That's the difference. Okay, which of these is the time taken for this person to recover from the exercise? So when the person is recovering, that means that their heart is returning all the way down to their resting heart rate, which should be back to 72. Right, so their maximum heart rate here reaches, so at 20 minutes, that's when it's at its maximum, and then it takes them until minute 29, right, this minute here. Oh, is it 29? Yes, for the heart rate to go back down to its resting heart rate. And so 29 minus 20 is 9 minutes. 29 minus 20, that would be 9 minutes. All right, complete the passage about what happens in the body during exercise using the correct words. So during exercise, gas moves from the lungs to the blood. Okay, when it comes to the lungs and a gas moving, there's only 2 gases that are moving in the lungs. That would be carbon dioxide and oxygen. But the gas that is moving into the blood would be oxygen. Okay. Moves from the lungs into the blood by a process called, so oxygen moves from the lungs to the blood in a process called diffusion. Spaced out there. Diffusion. So diffusion, remember, is a passive transport. So diffusion is when molecules or when substances go down from a high concentration region to a low concentration region. This gas is needed by cells for what kind of respiration? That would be aerobic respiration. So there are two types of respiration, aerobic and anaerobic. Anaerobic is without, whereas aerobic is with oxygen. Also, during exercise, the amount of urine produced something as water is lost in the sweat. So when we exercise, we produce a lot of sweat to cool down our body. And when we produce a lot of sweat, what happens is that the water content in our blood goes down. And in response to that, we want to conserve as much water as we can in our body. And to conserve as much water as we can in our body, we need to make sure that we produce little amounts of urine. So that would be decrease here. Oh, why did I spell that wrong? Decreases. Okay. Next part. During exercise, body cells get damaged. Body cells are replaced by a process called mitosis. So this diagram represents one body cell but only shows 6 of the 46 chromosomes. Complete the diagram to show the new body cells. Okay, so mitosis is a type of cell division in which you form genetically identical cells. And because it's genetically identical, then we would just copy the number of chromosomes into the new cells. So here, I'm just going to draw this. Probably draw this as well. And then draw, that's 1, 2, 3, 4, 5. That's 6 chromosomes, right? So we'll draw 1, 2, 3, 4, 5, 6. So for this, the shape doesn't really matter too much. And then let's do another one here as well. Let's draw 6. 1, 2, 3, 4, 5, 6. All right. All right, last part. Mitosis takes place in 4 stages. Which of these gives the stage of mitosis where the chromosomes align at the equator of the cell? So this would be metaphase. This is how I personally remember it. Metaphase starts with the letter M, and so does the middle. The middle starts with the letter M as well, and the chromosomes line up in the middle. And we're done with question 2.
Alright, let's start with the third question. So it says, the diagram shows some equipment that can be used to investigate the energy content of different foods. So here we are given these equipment, and we need to describe how the equipment can be used to investigate the energy content of different foods. Okay, so I have included, so this is not part of the question, but I have included a diagram of what that experimental setup would look like. Let me just erase this. Okay, so basically what we have is the food here that's mounted on the needle, and you would burn the food, and when you burn the food, that releases a lot of energy, and that heat energy is being transferred into this water here, which will cause the water to rise in temperature. So the higher the energy content in that food, the greater the temperature rise. Okay, so first of all, the first thing that we need to do is actually put water into the test tube, but we want to make sure we have a fixed volume here, and we would use a measuring cylinder to do that. So you don't have to put numbers here, but I'm just putting it for clarity's sake. So use a measuring cylinder. To measure out the volume of water, then place put the water in the test tube. All right. The second step, so once you put the water into the test tube, we would measure the temperature of that water. So measure the initial temperature of the water, and we would do that with a thermometer. A thermometer. Okay, so once we do that, we would place the test tube into this clamp that we see here. Oops, I dropped my pen. Okay, so then we would place the test tube to a clamp stand, and then we would stick the food onto this mounted needle here. So place the food on the mounted needle, and then we would burn with a Bunsen burner. Alright, so once we do that, we would place the mounted needle right below the test tube so that the heat from the food can be released and would be transferred to the water. Once we do that, we would then measure the final temperature. Of the water. So the greater the temperature rise, the more energy content is being stored in that food. Alright, the next part, name two variables that need to be controlled in the investigation. So if you look back at the experimental setup, one of the things that needs to be controlled is the volume of water that's in the test tube. So let's write that down. Volume of water. Another thing that needs to be controlled is the mass of food that you put on the mounted needle. So you can add that as well. Mass of food. Another thing, so I'm just going to write 3 here. Another thing is you want to ensure that the distance between the test tube and the food stays the same throughout your entire investigation. So you want to have the distance be fixed. So the distance between, okay, make sure to not write beyond this line, between the food and test tube. Alright, last one. For this part, give 2 safety precautions needed for this investigation. Now, because we're dealing with burning food, we want to make sure that the burning food is not close to our body. So you want to keep the burning food at arm's length. So keep burning food at arm's length. You also want to make sure that you wear goggles for this experiment. Another thing that you can also add is make sure you don't ingest the food. You shouldn't be ingesting the food in the lab to begin with. Alright, let's do part B. Different people have different energy requirements. The table shows the daily energy requirements in kilocalories of males and females of different ages. Alright, describe the pattern. Okay, so when it says describe, we're just looking at the trends that are in the table. We're not explaining why that is. So we're just trying to look at the trends here. Between males and females, right? So let's compare the two columns here. If we compare these two values, we can see that this is larger. If we compare these two, this is larger, these two, larger, these two, larger. It's pretty much larger for males versus females, right? So overall, males have a greater energy requirement than females. Than females. Oops. Okay. Another one, let me just erase this. We could also compare in terms of the ages. So let's compare this entire column here, right? If we look at the younger males from age 19 to 24, they require about 2,700 kilocalories. It's 2,700, 2,600. You can see it's decreasing, right? 2,500, 2,500, 2,300, 2,200. It's decreasing. Let's see if the trend is the same for females, right? 2,100, 2,000, 1,900, 1,800. Again, you can see there is a decrease as we get older. Oops, let me just start with a capital letter for both males and females. The energy requirement, oops, my handwriting, requirements decrease as age increases. Alright, let's look at the second part here. Which of these gives a reason for the difference between the energy requirement by 25-year-old males and the energy requirement by 25-year-old females? So we know that for males, the energy requirement is greater than for females, and the only reason that fits with this, that can explain this, is that males have more muscle mass than females. So more muscle mass means greater amounts of energy required for those muscles. Alright, let's do the last part of this question. Calculate the percentage difference in the energy requirements of males compared with females in the age range 35 to 44. So let me just erase this so we can properly see this. So ages 35 to 44, that's 2629 for males. Let me just write this down. 2629 for males, and for females that would be 2,103. Sorry about that, 2,103. Okay, so to find the percentage difference, we would need to find the difference between these two values and divide it by the value for women. The reason why we're dividing it by the value for women is because it says compared with females, right? So the energy requirement of males in comparison with females. So you would find out the difference between these two values divided by the value for females times by 100 because it's a percentage, and we should get 25.